but some of the concepts might be slightly new to you. So just stop me if you need me to explain more about these topics. So in the last class, I was basically just introducing you to the idea of molecular biology and gone through the key seminal experiments that had happened. And then I started to introduce you to some basic terms. We talked about what are the constituents of a cell, how these constituents are interacting with each other, what are cellular pathways. And then I ended with some terminologies. So I'm repeating those terminologies. A genome is an organism's all the genetic material in the organism. So genetic material is something that is passed on from mother to the daughter cell, or from one organism to the its children. A gene is a discrete unit of hereditary information that is located on a specific location in the organism's genome. So if an organism has multiple chromosomes, so it will be located on a specific chromosome, and a gene is consisting of DNA. Genotype is the genetic makeup of the organism. So basically, if you were to compare two organisms, a genotype would be the unique set of DNA molecules or DNA letters which sort of identify that organism. Phenotype is the physical expression of traits of an organism. So, for example, again, I'm going with example that we started with earlier with Dave's example. If you are lactose intolerant, let us say, you might have mutation that allows you not to make the lactase gene as efficiently. So then your genotype would be the fact that you have a mutation, and the phenotype would be the fact that you are not able to make that gene and, let us say, digest milk efficiently. Nucleic acid is basically the biological molecules, the monomers that make RNA and DNA, and that allows organisms to reproduce. So a genome of an organism is again complete set of DNA. So one bacterium approximately contains half a million DNA bases. Human and mouse genomes are approximately 3 billion base pairs, right? So whenever we're talking about here base pairs, so if you talk about a bacteria having 500,000 or 5 lakh base pairs, you would be essentially talking about 1 million bases, right? Similarly, human genome has 3 billion base pairs or 6 billion bases. The human genome, the bases are organized in 24 distinct chromosomes, 22 are autosomes, and 2 are sex chromosomes. So this is just the same thing. So now if we specifically go back to look at DNA in detail, the letters, the monomers that construct the DNA are 4: adenine, guanine, thymine, and cytosine, and they have certain base pairing rules. So adenine base pairs with thymine by forming 2 double bonds. Cytosine base pairs with guanine, forming 3 double bonds. So cytosine and guanine binding is slightly more stronger. So this is how this base pairing rule looks like. This is the phosphodiester backbone, because of which DNA is largely negatively charged. It's something that we talked about in the last class. So it has a phosphate, and then there is oxygen here. So it's 4 oxygen molecules. And then the bases that are attached here to the DNA molecule. So a DNA molecule, a nucleic acid essentially would have a sugar molecule that you see here. It's a pentose sugar. It's a five ring sugar. Then there's a phosphate group that forms a backbone that's attached, and then there's a base which is shown in pink here. And this bases, when they face each other, they have a lot of oxygens and hydrogens available to form hydrogen bonds, and that's how base pairing happens and how you have complementary DNA strands. So DNA is always red from 5 prime to 3 prime orientation. So when we say 5 prime to 3 prime orientation, this is typically going along the phosphodiester backbone. So this is the 5 prime end of the phosphodiester backbone, and this is the 3 prime end of the phosphodiester backbone. So when new bases are added on the DNA, they are added at the 3 prime end of the phosphodiester backbone, and that's why typically whenever we say that DNA is being read, it is being read from the 5 prime to the 3 prime end, because that's how DNA replication happens, and that's how DNA transcription happens. That's how basically that's the nature of the code. Okay, so now again, this is how the information flows in the DNA. The DNA gets replicated to make more copies of itself. The DNA can get transcribed. So this information coded in the form of DNA is passed on into the form of RNA molecules, which is a transient copy of the DNA molecule. And then this RNA molecule can get translated according to the codon code into a protein. So the process of transcription in the context of eukaryotic cells, that's basically doing RNA synthesis. In the eukaryotic cells, that's happening inside the nucleus. And the process of translation, that is protein synthesis, is happening in the cytoplasm. In the case of bacteria, this happens, or prokaryote, this happens all over in the cell. So again, coming to the genotype and phenotype, genes are inherited and they are expressed in two ways. Um, so they you can either have the genotype, that is the genetic makeup of an organism, or you can have the phenotype, which is a physical expression. So in this case, if you see the phenotype here is the color of the eye, right? And the genotype would be specific mutations that are making an organism have that phenotype. So this is the codon pattern. So this is the table which is used, which is something which is universal to life. The table that is so these are the coding the sequence which is used by our different cellular machinery, specifically ribosomes, to read the RNA molecules and translate them into different 20 amino acids. So here, these are the amino acids. So in the last class, I mentioned that there are 20 amino acids initially, but now there are 21 amino acids recognized. Can somebody tell me the name of the 21st amino acid? You can Google the answer.
Selenocysteine.
Exactly. So selenocysteine is the 21st amino acid, and the way that the reason why it was recognized was because it was found in lots of organisms at significant abundances. Okay, so slightly going deeper into how genetic materials work, that is, how does the DNA work? So I'm going to go into the few prior experiments, going to talk about what are the different kinds of mutations in the DNA and how the DNA itself is organized in the form of chromosomes, genes, and how these genes are ordered, so on and so forth. Okay, so going back, Mendel was the first person which who was able to show. He was an Austrian monk. He was able to show how parents pass on traits to their children. He did that by crossing pea plants. So your mutter, right? Plant, the green pea plant. So typically you have green pea, but the pea plant can come in various varieties. You can have pea which is green, which is yellow, and they cross. So then he was basically looking at these kinds of physical traits: color of the pea pod, the height of the plant, the number of pea pods inside each, each vessel of pea. And then they were basically, he was saying, okay, if you cross two plants which are very tall, would you expect uh, in what ratio would you expect the progeny to be tall? I mean, if if you see that, okay, all the tall people when they have babies, they are always tall. Is that the case? That's not actually the case. There is depends upon what kind of trait height is, right? So that's how he discovered the question that do traits come from a blend of both the parents or only from one parent, or are there some rules around it? So for example, when he crossed a purple plant with a white plant, typically the plants, most of the offsprings were purple. So you could say that the trait purple is a dominant trait and the trait white is a recessive trait because it gets dominated by another color. So next, it was basically DNA was then discovered to make up the genetic material which was inherited in between organisms, and this is just a repeat of what I said earlier. So now, when you inherit DNA during the process of DNA replication itself, and during the process of inheritance, there are certain mutations that are coming up. And this is how it looks like. So if you have a normal DNA sequence as shown here, you can have a mutation which changes the code of DNA, and as a result of which, say, a T here originally gets replaced by a G, right? So now what happens when the DNA gets mutated? If the DNA gets mutated, there are some mutations which are good. So for example, there are mutations that cause sickle cell mutations in a gene called the sickle cell gene. That's something which is prevalent in a lot of people in Africa who have lived through malaria for a long time. So these mutations make the red blood cells of a specific shape, and when they have sickle cells, then the malaria is not able to um, is not able to replicate very well inside these kinds of red blood cells, and therefore the people who have sickle cell mutation are actually protected against malaria. So this is actually in some ways a good mutation. There are bad mutations. Bad mutations are essentially mutations that cause traits that are harmful to the organism's function. They can be sometimes fatal. So, for example, Huntington's disease, there is a basically a mutation that causes the degeneration of new nervous system. So in specifically in Huntington's disease, you have instead of changing of letters, you have insertions of large number of DNA material, which leads a gene to not work properly, and because of which the nervous system goes bad. And there are certain mutations which are simply not making any difference in the organism, and this is typically happening because of the codon sequence. So we know that when an RNA molecule is being translated into a protein molecule, it is three bases which are translated, right, right at a time. And the third base is called the wobble base, because very often the identity of the amino acid is can be already ascertained, can already be mapped by looking at the first two nucleotide sequences. When we talk about mapping them to a protein, mapping them to an amino acid. So next, it was found that genes are actually organized physically in a linear fashion. So they are linked to each other, and this was found by Thomas Morgan, who was doing experiments with fruit flies, as I was talking to you about. This was the genome that was also sequenced immediately, released immediately before the human genome was completed in the year 2000s. Okay, so DNA is basically, if you look at the organization of DNA, it's a double-stranded helix, right, with the reverse and the forward strand going like this. The DNA itself, it needs to be, so if you were to take the DNA in your own cells, from the human cell body, if you were to take the entire DNA fragment and you were to stretch it out linearly, it would be as long as 1.2 meters. So each cell contains DNA, which is equivalent to 1.2 meters long, right? So imagine when you take the DNA from all your cells in your body, actually the amount of DNA that you have would reach from the Earth to the moon approximately 8 times. So all of that information is fit in your entire body, entire. So 1.2 meters of DNA is fitting inside the cell, and this is done by a compaction of DNA. So basically, you can imagine that if you have a string and you keep on rotating it, it starts to sort of get compacted. And this is in the cell, this is facilitated by interaction of DNA with certain kinds of proteins called histones. Because DNA is largely negatively charged, histones are proteins which have very positively charged amino acids, so they bind very tightly to the DNA and help the condensation of the DNA, basically the contraction of the DNA into much smaller form. And typically, you see that DNA is shown in the form of this kind of X, this chromosome. This is the shape of the DNA when the chromosomes are separating during the mitosis process in the metaphase of the cell cycle. So I hope these terms are known to all of you. Again, right? Show
Yes,
of
sir.
hands, how many people know
Yes,
metaphase?
sir. Yes, sir.
Metaphase, chromosome, interphase. Quick show of hands, please. If you're sleeping, then this is this is how I know who all is sleeping and who all is listening to me. Okay, so I can see that there are seven, eight. Okay, about 8 people who are listening to me. 10 people. Okay, that's great. I have a good audience today. 13, 14. Okay. Come on, let's 18 people are attending, so 5 people are not listening. Okay. Okay. Mayank, Satyakam, Abhishek. Okay. It's probably too early in the morning for you guys. All right, so please keep your hands down. So yeah, so this is how the DNA is condensed. And when you take a look at how the DNA is condensed in different organisms, so when we talk about most bacteria, please keep your hands down. They are having a single chromosome. It's a single circular chromosome. So the DNA is, yeah, it's circular in nature. When we talk about different eukaryotes, so the first organism that was sequenced, yeast, that has about 17 chromosomes. The total base pairs is about 12 million, 13 million, sorry. When Drosophila I was talking about, it has about 160 million um, uh, base pairs, which are organized in four chromosomes. Humans have 2.9, 3.3. These things keep on updating, and these are organized in 23 chromosomes. and say corn, which is called Z maize, which is like the bhutta that you eat, right? On the street, that's about 5 billion base pairs organized in 10 chromosomes, right? So there you see that there is some kind of like number of DNA base pairs often don't correspond to the complexity of the organism. So you can say that definitely, even though a corn has many more DNA base pairs, it is most likely much less complex than humans, who have about half the amount of DNA base pairs. And how this complexity is achieved is something that I'm going to talk about through yeah, in the coming slides. So what do genes do? So genes basically are making proteins, and this was This was shown by a very interesting, very easy experiment in early 1940s in Stanford, where researchers were working on the bread mold. So if you keep bread for a very long time in your fridge, you would see that mold will be growing on the bread. So it was the same in this mold is called, it's basically a fungus. It's called Neospora, right? So what researchers did was they thought that this mold, um, I mean, it's easy to grow, so why not research with it? And what they hypothesized was that because this mold always happens, right? There must be some property in the mold which allows it to eat up the bread, right? And that it must be doing this through some enzyme. So what they did was they thought that, okay, if we were to make mutations, and if this is one specific mutation that is in one specific gene which is causing this mold to eat up the bread through this enzyme, then we would be able to prove that there is one gene which is responsible for one mutation and for one enzyme. So what they did was they grow the mold in two kinds of media. One media is called the complete media, where you basically make the organism happy. You give it everything. So it's uh, like if you were to compare it with the human terms, you would have a person living with all the amenities, right? Living in a, I don't know, like a five-star hotel. And then you have organisms which are grown in minimal media. So they are grown like, I don't know, somebody who's below poverty line who has just enough to survive, right? And what they did was they irradiated this organism, Neospora, to induce mutations. And as a result, they got different kinds of mutated spores. It's basically single species, a single individual of a fungus is often called a spore, and they placed this spore into the minimal media. All right, so that basically they did not have things that you would expect in you would expect in on the bread, right? So what they showed was that when they exposed it to X-rays, the growth and like the spores started to have like a specific shape, right? And what they did was they were able to then uh, replace different kinds of vitamins, amino acids, and nucleic acids in the medium, in the minimal media. So because in the minimal media, the organism has to do everything by itself. So the pathways which are sort of utilizing vitamins, amino acids, and nucleic acids themselves, the organism would be making it by themselves. And now, if you were to cause the mutation that allows the organism, the pathway inside the cell or the gene which is making these things for the organism, which are important for the organism, if that pathway is mutated, then the organism will not be able to survive. So what they were doing was they added different kinds of amino acids. They removed different kinds of amino acids from the minimal media, and they did this mutations, and they supplemented the medium with specific amino acids. And what they found was that Neospora survived when it was supplemented with vitamin B6. When X-rays damaged genes that produced a protein responsible for synthesis of vitamin B6, then it had to be supplemented with vitamin B6. And similarly, they isolated three mutant strains which were unable to synthesize vitamin B6, B1, and another organic compound, which were found to be essential growth factors, right? So now they were basically able to show that when you cause mutations, specific phenotypes or ability of an organism to metabolize or produce a specific vitamin is removed. And by this, they were basically able to show that there would be specific genes that are probably responsible for it, because when they mated a mutant strain with a normal strain, right? That was basically differing by a single gene. This problem of not being able to make vitamins was going away. So essentially, um, there is more than one step in the system with synthesis of vitamin B6, and the mutation affects only this one specific step. All right. So by mating, this mutation was removed. So, going to how the genes make proteins. So once they had people like the Beetle experiment for establishing how the proteins are made, people started to look more deeper into how proteins are made. And here, actually, seminal work was done by Hargobind Singh Khurana, who is India's one of the India's Nobel laureates, who basically they were taking small repeated RNA molecules, and they were seeing how what kind of amino acids they were synthesizing. And by very elegant experience experiments where they were taking the same repeating code of amino acids like 80 80 80 in sorry, same code of nucleic acids 80 80 80 in the RNA molecule, or GC 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 in the RNA molecule, they were able to now obtain just the same amino acid when the cell was translating this code, this mRNA molecule, into a protein. So they were able to then specify that there are 20 amino acids and in different proteins at that point of time. Now that has been updated to 21, and these amino acids basically make up all the proteins, and the proteins are essential to most work inside the cell. So they form structural part of the cells. They form enzymes that digest nutrients. They execute specific metabolic functions as molecular machines, and they obviously mediate the flow of information from within a cell to among the different cellular components and between cells as well. Okay, so this is just some information about DNA, how it was discovered, th- and yeah, how it looks like. Yeah, this is just going deeper into DNA. This is just repeating what I said earlier. So typically, when you have DNA itself, it's negatively charged, so you can look at it on a gel, and this was essential to be able to do DNA sequencing, because you had to isolate different fragments on a gel. I already talked about hydrogen bonding, DNA protein interactions, major grooves and minor grooves, and DNA polymerization, which happens from 5' end to the 3' end because of the phosphodiester bond. And because of this idea of DNA being going from 5' to 3', people were able to figure out how DNA replication would be happening, or how DNA would be copied from one organism to the other organism, passed on, right? So essentially, this is how it's happening. When the DNA is getting replicated, you have uh, you have a bubble formed. So basically, the double-stranded DNA is unwound, and when you are unwinding the double-strand DNA, there is polymerase, which is polymerizing DNA. So it's called DNA polymerase. So what the DNA polymerase does is it uses the uh, the template, one DNA strand as a template, and goes from the 3' end to the 5' end, because it is the DNA polymerization happens from the 5' end to the 3' end. So this particular strand is called the leading strand, right? And it keeps on going, keeps on unwinding the bubble, and the DNA replication goes happens in the basically in this direction. And on the opposite strand, because the DNA replication can only happen from 5' to 3', so when initially this part of the DNA would have been opened, you would have the DNA replication happening from 5' to 3', and so basically going in this direction. And as and when more and more parts of the DNA are unzipped, you see more and more kinds of more and more such fragments come up. These are called Okazaki fragments, and they eventually then are joined by what's called DNA ligase that joins these fragments from the 3' end of one DNA fragment to the 5' end of the other DNA fragment. So this is going to be the first set of bioinformatics questions that we'll be asking. So how does the cell actually know actually where to can we use this information of DNA replication to actually find out how does the cell know where the replication should start. So typically replication starts at what's called origin of replication. So the first question that we can ask using information about DNA replication of how the process proceeds is by asking the question exactly where does the cell begin the replication. And this is going to be in the module 2 that we are going to take up in the next week on most likely Thursday or Friday. All right. I already talked about the superstructure, so I'm not going to talk about that more. So this allows the DNA to be packed. And because the DNA is packed, there have to be certain mechanisms through which the DNA has to be unpacked when its copies are being made. And this unpacking is happening through transcription factors, which go and bind to the DNA, which are found in the 5' end of a gene. So I'll quickly break here and ask you guys, do you know of any transcription factor? Since you have done molecular biology last semester, do you know of any transcription factor? Do you know of any kind of signal where
Yes,
these
sir. Yes.
transcription factors might be found?
You
Yeah.
like in lycopyron and etc.
Okay.
Sir, in lycopyron and TRP-operon, there are transcription factors.
Okay. But is there a universal transcription factor which is required for transcription in the case of bacterial genomes, bacterial DNA?
So basal transcription factor.
What is it called?
Sir, like it is a type of a transcription factor. One means
There is a specific name for it. It's called the sigma factor, right?
Sir, I don't know, sir. We
Okay,
have
you
not
were not
learned
taught
for
this.
it.
yes, sir. Yes, sir. It was taught in the microbiology class.
Yeah, so this is sigma factor, right? So do you know how does the sigma factor, where does the sigma factor bind? Like how does it know where a gene lies on in the genome? How does it recognize that I have to bind here on the DNA? Have anybody of you heard of Tata box? TATA box?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yeah, so can somebody tell me? So maybe Ashi?
So the Tata box is a sequence. I do not remember at -10 or at -35, but it is a definitive sequence that is TATA AA,
Uh-huh.
and this sequence is recognized by the transcription factor, or the proteins recognize have a site for recognizing the sequence. They bind there and start the transcription from there.
Exactly. So now, basically, just like this one, so this is a transcription factor that is required for, like, it's called a basal transcription factor, but just like that. So Tata box typically lies between -10 to -35, and then there's also something called the Primnov box. So like just like that, there are a lot of boxes, right, which are nothing
Sir,
but.
it doesn't like it doesn't lies from -10 to -35. It perhaps lies at -10 or -35.
So can you
Like
confirm?
I don't remember, sir. I have to search that.
Can you search? So when you're correcting me, it's good that you search, right, and tell me. So I'm waiting. So it's -10 or -35. And where does Primnov box lie? Are you telling me, Ratnesh?
Sir, Tata box is -35.
Okay. And -10?
So that would be the Pripnovox, that is TTGACA.
Okay, so that's good. So basically, this is the third module that we'll be talking about. Is that most of these sequences, when you say TAAA, this is not like a exact sequence. So different organisms have different kinds of Tata boxes. So and for different sets of genes, you have different kinds of transcription factors. So these are the sigma factor basically is a basal transcription factor. There are certain transcription factors that are unique to certain set of genes. So for example, there are transcription factors, let us say in the lac operon, right, which are specifically binding to the lac. What in the lac operon you have the lac A gene, right? What is the activator in the lac operon? Can somebody tell me?
So lac operon is an operon and it regulates lactose metabolism inside the E. coli cell.
So what is the gene that it regulates?
Sir, there are 5 genes, 3 genes
It's a
there.
lag Z, right?
Lac Z, yes.
Lac X, Y, and Z. G.
Okay.
Yeah.
So when you talk about lag Z, typically that's what you are taught, right? So in the 5' end of the lag Z, what is binding? Is it a
What is
specific
binding?
transcription factor binding, or is it a transcription factor binding? Is it a repressor binding? What is binding?
Sir, is
I
it,
think it's a
it
lack
a
repressor.
repressor binds like, yeah.
Ashi, you had an answer?
Yes, so the lack repressor.
The lac repressor binds,
That is
right?
called allolactose, perhaps.
Perhaps, or are you sure?
Sir, I have forgot all these things. So,
So that's what this is what this lecture is for. So maybe you can quickly Google that as well. So what I meant to say here is that in the lac operon, you have the LAC-Z gene and you have the specific lac repressor in this case. Just like that, you have transcription factors that activate transcription. You have transcription factors that repress transcription as well. But all of them have this unique feature or ability to recognize specific sequences. So in module 3, we are going to talk about how does the cell recognize specific sequences. And now when we talk about recognizing specific sequences, it's not always the exact sequence that is recognized. This sequence has some mutations here and there because you cannot always have an exact sequence that the cell wants to recognize. So can somebody tell me? So for example, the TATA box, Tata box, why would you think that it could have certain mutations? Why would the cell not want to always recognize the exact TAA box and would want to tolerate some mutations there? This is nothing to do with what you have learned. I mean, it's just something to do with what um, something that I've already talked about in this lecture so far. So why would you not want an exact match always? Would that have a problem inherently when the cells are dividing?
So I guess we've
sir,
learned
because
about this in terms of a good promoter and a bad promoter. So a good promoter sequence would lead to production of multiple copies, but in the case of bad promoter, the copies would be lesser. I do not remember the exact terminologies, but when there are mutations in the Tata or Pribno boxes and the spacer sequence between them, when it's not of the ideal length, at that time, the number of copies or the number of transcriptions that take place are comparatively lesser. So
Okay.
maybe this is a mechanism to regulate the number of transcripts that we obtain.
That's true. But what if just by chance you
yes,
have a
sir.
mutation? Like mutations are randomly happening, right?
Yes, sir.
So do you think that if it recognizes something suboptimal, I mean, not an exact sequence, but there is some mutations tolerated, right? Then you can have, there is more chance that mutations, I mean, the organism is not going to stop transcribing, right? Or stop making genes or stop doing the transcription process. So that's actually what happens. Because there are mutations all the time accumulating, you don't always have a very exact sequence that is recognized for most transcription factors. So for basal transcription factors, you have the TATA box, but the TATA box also is, it's not just like a standard entity. You have also some kinds of like, it's not just TATA. Every organism has its own TATA box sequence, and the TATA box sequence also itself has certain mutations that are tolerated. Sometimes it is to do with changing the transcript amounts. That's definitely true. But sometimes it also has to do with the fact that you would like to tolerate mutations that happen just by chance. And so this is what we'll be looking at in the module 3, how transcription factors actually recognize these these motifs, or also called transcription factor binding sites, which are not always exact, and how bioinformatically can we actually search for these sites inside the genome, and how we can annotate them. So this is the third module that we'll be having. So going further, basically coming to another very interesting part of transcription process itself, which is splicing. So how many of you know about splicing? Was it covered?
Sir, it is not
So
covered.
in brief, so in like very brief.
Very briefly. Okay. So when I said that maize or a corn plant has more genes or more DNA content than humans, but still the humans are seem to have more complexity than these organisms. That's probably driven by the process of splicing. So process of splicing basically allows the same gene to be stitched together in different ways to make different kinds of proteins. So in this sense, you can think about a gene to be composed of different Lego blocks. You know, you have the Lego, small blocks, like one of those games. And when you make the blocks, you put the blocks together in a certain order, then you have a different kind of a structure come up. So the same thing happens with proteins as well. So you have your same thing happens with the spliced RNA. So this is your double-stranded DNA that makes a single-stranded mRNA molecule. This mRNA molecule now would go and it will translate, and you'll have a protein, right? This is the standard process. But now in the case of splicing, what happens is, let me go to the splicing part. Yeah. So in the case of splicing, what happens is that your initially the what RNA molecule is getting transcribed, that gets chopped up into smaller pieces, and you can, from the same RNA molecule, you can actually make different kinds of final transcript messages. So for example, in RNA molecule, so the parts which get chopped out are called introns. So these are parts which do not code for a protein, and the part which code for a protein are called exons. So here in lighter color gray. So now when the introns are getting chopped up, sometimes just by chance, not just by chance, by in a regulated manner, you can also chop up not just this introns, but you can also chop up an exon in between, right? So for example, here what you're seeing is that there are 3 exons in this gene, and when the introns are chopped up, you finally get a protein message which has 3 exons put together, exon 1, exon 2, exon 3, which forms your messenger RNA, which would finally get translated. Now, just by chance, you can have sometimes when introns are getting chopped up, your exon 2 might also get chopped out. So as a result of which, you will get a protein which is going to be missing exon 2. So now you are physically making proteins that are missing parts, right? Or having sort of combining different parts of different exons. So when you do something like this, you would essentially give rise to multiple different versions of the same protein. And this allows humans, for example, to rapidly expand the same set of genes into many different kinds of proteins. So typically each human gene makes approximately 10 to 50 different kinds of proteins depending upon the splicing pattern. So for example, if you have n exons, right? So each exon can either be finally be present in the final message, or they can be absent from the final message. So in principle, if you have n exons, you can have 2 to the power n different kinds of combinations of these exons, right? I hope everybody follows this. Yes?
Yes, sir.
Yeah.
Yes, sir.
So yeah,
Yes, sir.
so basically you can have 2 to the power n different kinds of combinations, and therefore you can make 2 to the power n different kinds of proteins from the same code. This is something which is specifically happening in the case of higher eukaryotes, such as humans, who have a lot of complexity. All right, so I think I'll just like skip this part. You all know what is a tRNA is. tRNA is a transfer RNA, which has this
Yes.
anticodon part, which recognizes the codon in the RNA molecule, and accordingly, each tRNA is having specific amino acids attached to it. So if you have 20 amino acids, you have 20 tRNAs, right? Is that a correct statement? Are there 20 tRNAs?
Yes, sir.
Perhaps no, sir.
Okay, so this is a question that I'll ask you guys in the next class. Maybe you can find out. So there are 64 codons, right?
Yes,
If I
sir.
look at the codon table, there are 64 codons.
Yes, sir.
So how many tRNAs would be there?
So 64.
Are you sure? 64 tRNAs? There are some codons which don't
No,
code
sir.
for a. So find this out, okay? So this is something that you can find out. We can quickly talk about it in the next class. So there are 3 stop codons, right? So
Sir,
they
I just
probably,
checked. It's written there are 31.
right? So this is an interesting question that you can answer in the next class. Why are there more tRNAs and why are they not 61 tRNAs and why only 31 tRNAs? Okay, discuss this. All right. So these are just the terminologies. mRNA is mature RNA. It's basically the final message that is getting translated. So bioinformaticians basically say RNA, they basically mean mRNA. tRNA is the transfer RNA, which is basically transferring genetic information from mRNA to the amino acid sequence. And you have rRNA, which is ribosomal RNA, which forms a part of the ribosome. So these are RNA molecules which are actually structural in nature. Then you have heterologous nuclear RNA, heterogeneous nuclear RNA, which is basically the form of the RNA molecule which is made before it is spliced into the mRNA molecule. Finally, phosphodiester bond you already know about. Promoter is basically, it's the special sequence indicating the starting point of RNA synthesis. So promoter is essentially the region where transcription happens or begins. So terminology for transcription would be RNA, which is ribonucleotide. It is basically a UGC. Instead of a thiamine, you have a U as uracil. Um, and these are because it's RNA, so it's not deoxyribose. This is ribose sugar, so it has an extra OH group, and having an extra OH group in RNA makes it more volatile. So it's a more temporary molecule. Most of the RNA, the process of transcription is basically polymerizing different RNA bases, RNA monomers. So therefore, the enzyme which does this is called RNA polymerase because it polymerizes RNA molecules. There are different kinds of RNA polymerases which are transcribing different kinds of RNA molecules. So typically, most of the protein coding genes, the genes which finally make proteins, are transcribed by RNA polymerase 2. RNA polymerase 1 and RNA polymerase 3 transcribe tRNA and mRNA, right? Which are basically RNA, which are functional or structural RNA, whose main job is not to actually make proteins. And then there is something called terminator, which is a signal which halts transcription. So I guess this is something that you all guys know as well. What's a
Sorry
Yes,
terminator?
about
sir.
RNA 2, RNA 3. These things I don't know. So perhaps
Okay.
we have not learned.
Okay. Terminator. Do you know about terminator in transcription?
Yes, sir.
Yes, sir.
So just like there is a promoter sequence, there is a transcription factor binding site. which allows the proteins to recognize where to bind and where to begin transcription. There are terminator sequences in the 3' end of the gene, which tell the RNA polymerase machinery to stop and stop transcription, right? All right, so this is all this part already covered. So translation happens through ribosomes. So typically you have an RNA molecule which will go and bind to the ribosome, and then the tRNA molecules as shown here, they will come, they will bind to corresponding 3 base pair codes on the mRNA molecule. So they have the anticodon, they will bind to the codon, and simultaneously they will keep on adding the amino acid, and these amino acids are going to get polymerized. So you basically, this process is called translation because you're translating, literally you're translating one language, one code. So you're translating ATG, or like you're translating AUGC into another set of language, which is basically having another letters, some other letters, which is like your 21 amino acid letters, right? And when you have the stop codons, you basically at this point of time you have the protein translation stops, and the amino acid, this polypeptide, this chain of amino acids, which is also called a polypeptide chain, because two amino acids are bound together by a peptide bond. This polypeptide chain is cleaved off, and then the protein folds, and you basically get the final product of the protein. So this happens at a much slower rate, approximately 10 codons per second. So when you say 10 codons per second, this is approximately 30 nucleotides per second that it's about reading. Okay, so these are the terms I already talked about. Anticodon, you know, C terminus is the 3' end of the protein, which is basically having a carboxyl group, COOH. N terminus is the 5' end of the protein, which has a free amine group, which is NH3. So basically, if you look at it, at the beginning of the protein, this part is the N terminus. At the end of the protein, this is the C terminus at the 3' most end of the protein. Okay. So this is basically how it happens inside a bacterial cell, because all these processes are happening simultaneously. You essentially have your DNA. The DNA gets opened up when a protein goes and binds to the transcription start site, and an RNA polymerase is then recruited to make a copy. So this copy is the copy in blue, this blue mRNA molecule. This blue mRNA molecule goes and it binds to a protein complex called the ribosome complex, which interacts with tRNAs, which basically are going to have which tRNAs have this amino acid bound to them, and typically when the ribosome keeps on moving along this mRNA message, these amino acids are joined together to form a polypeptide chain, and that's how translation happens, and the protein forms a three-dimensional structure. So this is sort of an open problem. Like until 2 years ago, if I was teaching this course, and when this course was being taught, typically you would say that you cannot predict the structure of a protein just by looking at the sequence of an amino acid, right? So if you have this amino acid sequence, so if you are given a gene sequence and you know exactly from where transcription is, from where translation happens, so you can basically look at the 3 base pairs, right? You can translate them into RNA, and then from RNA you can so you can first transcribe them into RNA. So basically you will make a complementary fragment of your gene into the mRNA space. Then from the mRNA space, you're going to translate into the protein space to make a protein, right? And once you know this, you will get a sequence of amino acids. So until so far, people were not able to predict the structure of a protein just by looking at the amino acid sequence, right, or the polypeptide sequence. But now you have last year, Google unfolded its deep learning machine called AlphaFold, which essentially it has learned how proteins fold based on thousands of protein structures that have been fed into this machine learning platform, where it can basically, given an amino acid sequence, it can actually fold it into the most likely structure it's going to take inside a cell. Yeah, so basically now you can essentially go from having amino acid structure to actually folding that protein and looking at it in three-dimensional space. So basically protein folding happens in a couple of different ways. So first you have some basic two-dimensional folds that happen in a protein. These are called secondary structures. So you have got the alpha helix and beta sheet. These are two secondary structures, and you have also unstructured loops. That's a third form of secondary structure. And these secondary structures come together. Alpha and beta helices come together to form more complicated three-dimensional structures. So basically, you have your polypeptide chain. It forms folds into these kinds of structures. Beta sheet and alpha helix, and then they form further fold into more three-dimensional structures. So this is called the tertiary structure or three-degree structure. And these three-degree structures come together to form quaternary structures. Basically, two or more tertiary structures come together to form the entire protein machinery, and that's the final functional form of the protein. Okay, so the last part is how the DNA is copied and analyzed. I think this is some part that you guys have probably done in the laboratory part of your molecular biology courses. Can I see a show of
Sir,
hands again?
we don't have laboratory course.
No, but like you must have known about PCR, cloning, restriction
Yes, sir. Yes, sir.
enzymes.
Yes.
Yeah, can I please show of hands because this is very important. Okay, so basically there are, because the DNA is complementary, you can use the same idea to amplify DNA through a polymerase chain reaction. You can also cut and paste DNA into a plasmid and can amplify that plasmid. This is called cloning. And you can, because of the negative charge of the DNA, you can run it on a gel and you can basically resolve the DNA fragments based on their size. And this is basically called DNA electrophoresis. Okay, so now when we have these kinds
Sir,
of things, what kind of problems can we actually um, do? So bioinformatically, one problem that we can address is that if you have given a genome, you can actually cut it into small pieces randomly. You can do what's called Sanger sequencing. So you can essentially read these small pieces. And because you are randomly fragmented these pieces, you can overlap them together like a puzzle and you can assemble the genome. So this is one problem that we are going to take up in the module 4 of the course, as to if you are given a large DNA sequence, how do you fragment it randomly and then put it back together, and then can actually tell what was the original sequence that you were working with. Once you have done that and you know that your DNA molecule is organized in the form of genes that are linked together, so you can compare either the entire genome or specific parts of this genome that you have assembled, and that's going to be forming the next modules 5 and module 6, which is going to be comparing two sequences of DNA molecules and comparing multiple sequences of DNA molecules. And then after you have compared the multiple sequence of DNA molecules, we're going to talk about phylogenetic trees. It's going to come in the module 6 and module 7, where you'll be I'll be talking to you about how you can draw evolutionary histories of how molecules are related to each other by doing these kinds of multiple comparisons. And then yeah, so basically come to that part. And in between, I'm also going to talk about the Human Genome Project. I think this part I can skip. Yeah, this part I can just skip. I mean, this is just for reading so that you are all on the same page. So again, I'm going to end the class today by telling you that first, so this was the first part of the module where I basically told you why you need bioinformatics, because there's a lot of information and some basic ideas of molecular biology. I'm sorry I went through this very fast, but this was under the assumption that most of these things are known to you. So here, the lab, basically we're going to do it in the next class. The lab is essentially searching biological data on PubMed. So then the next class I'm going to talk about biological databases. So how all the information which is out there is organized in biological information, biological databases. Then I said the first question that we are going to talk about, then module 2 is going to be how DNA, how does the cell know where to begin DNA replication? So this is going to be giving you some DNA matching algorithms. Then we are going to talk about how does the cell know where to start transcription. So like Tata box or the lac operon, where you have where the lac repressor is binding. So here we are going to talk about DNA finding regulatory motifs or finding DNA regulatory motifs, algorithms to do that. Module 4, how do we sequence an entire genome? So we're going to talk about genome assembly. Then I'm going to talk about the genome project, which is finding the module 5, which allows us to understand the technologies behind genome sequencing. As I said, once we have the genome, we know that the genome is made up of sequences or it's made up of genes. So in the next module, I'll be showing you how we could compare short fragments of DNA through sequence alignment algorithms. Then we're going to extend this to multiple sequence alignment algorithms so that we can uh, visualize how multiple sequences might be related to each other. And once you know that how the different sequences you can match different sequences, you can organize them in the form of trees. And here we'll be using the example of SARS-CoV-2, which is COVID-19, and we'll try to do some detective work as to which organism actually gave us COVID-19 through this kind of evolutionary trees. In the second last module, we're going to talk about, now that you have all this information, all these DNA molecules that are out there, all this information that is generated, is there a quick way of actually searching for DNA fragments inside large databases? And here I'm going to talk about the basic local alignment search tool and the algorithm behind it. It's an ingenious algorithm, and these fall in the category of heuristic algorithms. So module 6 and module 7 are typically, the sequence alignment problems are dynamic programming algorithms. And in the module 10, I'm going to talk about how do we locate a disease causing mutation. And here I'm going to talk about basically introduce the algorithms called combinatorial pattern matching algorithms. Again, so yeah, I hope I can see most of you in the upcoming lectures. I assure you it's not going to be as boring from next week onwards, because we're going to talk, hopefully I'll be talking about a lot of new stuff for you, right? I'll stop here at this point of time, and this lecture has been posted on Moodle already. I hope everybody can access it. Can I see a show of hands of people who have tried to